I'm going to be talking about active transport, more specifically the experiments that led to the discovery of active transport. Ultimately, there were 3 experiments that led to the full discovery of what active transport is, but the one I'm going to be focusing on is the one by Dennis Robert Hoagland, and he set out to discover the ability of plants to absorb salts against their concentration gradient. And ultimately, in the end, what he discovered is that plants depended on nutrient absorption and translocation on metabolic energy, and he did this using model systems. But first, I want to talk about the 2 kinds of active transport. There is primary active transport and secondary active transport. With that primary active transport, the sodium-potassium pump moves the sodium using the energy from ATP hydrolysis, which is shown down here, the ATP separated to ADP in an inorganic phosphate, to establish a concentration gradient of Na, where there will be a higher concentration of sodium on one side of the membrane compared to the other. Then there is secondary transport, active transport, and that would be when the sodium moves with the concentration gradient that was established by the sodium-potassium pump, and now it drives the transport of glucose, which is this little green molecule, against its concentration gradient. So as you can see, that sodium is coming in and it's forcing that glucose to go across the membrane and go against its concentration gradient as it is moving to an area where it's already higher down here. So at this time, it wasn't understood. People did not know how the plants were able to absorb these inorganic elements. And so at the time, it was thought that they were doing this through osmosis, antagonism, or possibly the permeability of the plant. But none of these could really explain the absorption or the retention of the concentration gradient. So Dennis Robert Hoagland wanted to explain the phenomenon of potassium accumulation in plant cells, and at the time this was thought to be created, these ions were thought to be created in the vacuole. So what he did is he selected freshwater algae, which tend to have relatively pure sap, vacuolar sap, which could be obtained. And so what that is in the vacuole of plant cells, there is this little area right here, which is the vacuolar sap. And what this is basically is just the fluid that exists inside the cell vacuole, and its job is to store resources, and it also helps to sustain the plant mechanically. And so that's why he wanted to look here to see if the ions are here, and that's where they were being absorbed. For example, he wanted to test the absorption and accumulation of bromide, which could take place only under metabolic processes. And this was his ultimate goal, was to determine if it was a metabolic process that was causing this absorption of salts. So what he did is he took lab-grown... Actually, first, let's talk about permeability. So as you can see in this figure, this is what he was trying to disprove, that it was simply just simple permeability. So what that is when something is highly permeable, stuff is easily moved through it. Things can pass through it very easily. So this one has high permeability as there's a lot of open spaces. It's not very tightly compact. Things are able to move very easily with low effort. Over here, where it is lowly permeable, it's very tightly compact. There's not a lot of open space, and things are going to have a very hard time moving through there. It's not going to be easy, and it's going to be very difficult. So to go back to those shooting roots, he made lab-grown plants that had these shooting roots, which he could later separate, and he wanted to test them under conditions to see what the salt concentration would be. So what he did is, among essential conditions that would normally create very high salt concentrations, he found that there was very low salt actually found, but high carbohydrates. But then in the excised roots that he found that it was the highest salt concentration highest salt accumulation that had thus been studied at the time. And then later, to set out more to prove that it was a metabolic process, he took barley roots and he studied those under specific conditions. And then he also wanted to reverse the external concentration gradient, which would influence the inhibitors and the ion accumulation. And he also wanted to use radioactive isotopes to help to separate the two stages. So the two stages being from a external medium to the cytoplasm and the cytoplasm to the vacuole. And then through this, it was able to be determined that it is indeed a metabolic process. And like I stated earlier, he discovered that the nutrient absorption from these plants and the movement of the nutrients was indeed a metabolic energy, and he did this using those innovative model systems of those plants that he created and using those radioactive isotopes.